The presence of small molecular weight hemoglobin species in previous generations of hemoglobin-based oxygen carriers have limited their clinical application. Technologies to increase the size of these materials will have a significant impact on the development of red blood cell substitutes. Hi, my name is Don Belcher, and in this talk, I'll be showcasing how a newly developed hemoglobin scavenging ApoHb-Hp complex may make hemoglobin-based oxygen carriers safer to transfer.
Before I begin, I would like to briefly state that myself, Ivan Pyers, and Andre Palmer hold a patent on several of the technologies related to the apohemoglobin and haptoglobin products that were used in these studies. So, why are we needing red blood cell substitutes? First, over the past decade, we've seen a 10% decline in the total donor pool. This, combined with seasonal blood shortages such as the recent COVID-19 pandemic, has led to widespread shortages of blood whenever it's needed. This, combined with limits on traditional red blood cell transfusions, such as limiting shelf life of only 42 days as mandated by the FDA, combined with blood type compatibility issues, has led to an urgent need for safe alternatives to red blood cell transfusions. Now, there's a lot of different materials that people are investigating, from perfluorocarbons to modified hemoglobins. Today in particular, what we'll be looking at is the polymerized hemoglobin. This is made through typically a glutaraldehyde or other polymerization agent to form large multimeric complex of the hemoglobin groups. But common among all these species that you'll see here is a need to increase the size of the material. Now, why are we interested in increasing the size? And this goes back to the function of red blood cells. So red blood cells' primary function is to hold the hemoglobin. If the hemoglobin is released from the red blood cells during hemolysis, it can freely extravasate through the endothelial cell junction, where it will then scavenge nitric oxygen. This nitric oxygen that's released by endothelial cells is a mediator in vasodilation of smooth muscle cells. Without it, we'll see systemic vasoconstriction and hypertension. However, if we were to transfuse a hemoglobin-based oxygen carrier with increased hydrodynamic diameter, this extravasation is functionally decreased or eliminated, which can lead to stabilization of the smooth muscle cells in the blood vessel. Now, this has previously been shown here, and as we decrease the size of the material, we see an increase in the mean arterial pressure. On the right, the material on the green line, the 100 kilodalton polymerized hemoglobin, contains a lot of small species, and when it's transfused at the bottom in red, we can see a significant increase in the mean arterial pressure after transfusion. So, in general, what we find is that increasing the size reduces the toxicity. However, it also can reduce the yield, increase the viscosity of the material, and lead to significant increases in processing time, which can potentially limit the application of these materials. So how can we clinically rescue these hypertension states? So typically what we can see is either use of nitrodilators to increase the nitric oxide production to relax the smooth muscle cells, or we can administer adenosine to target receptors to relax them. And both these methods are good, but they fundamentally treat the mechanism of hypertension. And if too much of these materials are delivered, we will shift the balance, and the patient will experience systemic hypotension, which can lead to negative side effects. So instead, we know the cause of the hypertension, which is the free hemoglobin. And instead of targeting the mechanism, we can target the cause. And to look at what happens normally under biological conditions to reduce these effects, we can turn to haptoglobin and hemopexin. Haptoglobin can functionally scavenge free hemoglobin, where it can be uptaken by macrophages. And hemopexin removes free heme that's dissociated from the oxidized form of hemoglobin. And both of these methods together typically work to detoxify hemoglobin when there's not a lot in the system. However, during large hemolysis events, or in the case of HBOCs, these are rapidly depleted. Now, a simple solution may be to just administer these proteins by themselves. However, both the haptoglobin and the hemopexin are very expensive. About $200 per milligram of haptoglobin and $600 per milligram of hemopexin. Due to this cost, and we may need grams of these materials to get any therapeutic effectiveness, it's unlikely that these current proteins would be commercially viable. So we went back to the drawing board and thought, is there any way that we could decrease the cost or find a substitute for these materials? We first wanted to consider what was making haptoglobin purification and the resulting products so expensive. So there's two general ways that haptoglobin can be prepared. The first is through purification of haptoglobin from cone fraction 4, a byproduct of the plasma purification process. Here, we could use either hemoglobin affinity chromatography or a combination of hydrophobic anion exchange chromatography. These methods either require a harsh dissociation reagent, which could effectively deactivate the haptoglobin, or large amount of resin. Alternatively, recombinant protein expression could be used. However, the haptoglobin has generally low yields due to co-expression of proteolytic enzymes. So instead, looking at method 1, we considered, is there an alternative to using column purification methods? And for that, we turned to a ultrafiltration methodology. So here, performed in 2 steps. In the first step, we sought to remove lipids from solution using a combination of centrifugation with fumed silica to extract a low lipid fraction 4 solution. We would then take this fraction 4 and purify it in a dye filtration train to clarify and concentrate the remaining compounds. This method was relatively successful, resulting in a haptoglobin product with approximately 93% purity. And this is a very cheap process to run due to the low cost and scalability of tangential flow filtration, with a current estimate of around $200 per gram to produce this haptoglobin product, which is significantly cheaper than the commercial products, which should enable much easier clinical application. The second material, hemopexin, was a bit more difficult. Here, hemopexin is only really available currently through recombinant expression, so we're looking at more of an alternative. And here, we thought apohemoglobin, which is hemoglobin without the heme, may be a interesting alternative. It's able to freely scavenge heme from solution, and it's potentially easier to produce. Previously, production of apohemoglobin was done either through an acid acetone extraction, which would sediment out the hemoglobin protein while extracting the heme in the acetone supernatant, or through methyl ethyl ketone liquid-liquid extraction, which would perform similarly with an organic and an aqueous layer. Unfortunately, both these methods have issues with poor scalability due to the solvents, two-phase separation, which can lead to protein unfolding, and finally, we need extensive dialysis and diafiltration to remove trace amounts of the methyl ethyl ketone or acetone to make it safe for administration. So looking at this, we thought, well, we can use tangential flow filtration again because we already need it for our diafiltration, and combine that with the extraction at the same time using what's shown here. So this is a apohemoglobin alcohol diafiltration. Here we combine hemoglobin with an acidified ethanol solution. This acidified solution allows us to extract the heme from the hemoglobin while in the same system, and then using a 10 kilodalton filter, we can extract based off of size, because the hemoglobin is about 32 kilodaltons whenever it's in the apohemoglobin form, versus the heme, which is significantly smaller in the hundreds of daltons. So we can use this method to efficiently purify large volumes of the apohemoglobin, and through it we actually get a much higher amount of active apohemoglobin. Here we define active as the apohemoglobin is still capable of binding heme. So in the alcohol-based purification, we get about 83% activity compared to 70% in an acid-acetone extraction. So now that we had both of these materials, we can next consider how do they interact with the polymerized hemoglobin. So we performed some initial experiments, first looking in the top row at different size-based effects. We know that haptoglobin binds to hemoglobin to increase its size, but we also found something similar happened with the polymerized hemoglobin. When they're combined together, we see a dramatic left shift in the size exclusion chromatography curves, indicating an increase in size. By looking at a deconvolution of these different waves, we can look at the polymer orders. And what we can actually see is a almost complete elimination of the 0th order polymer, which is the unpolymerized hemoglobin, and then a 50% reduction in both the first and second order polymerized hemoglobin, which overall leads to a massive increase in the size. The second thing we noted was not all of the haptoglobin was binding to the polymerized hemoglobin, as noted by the fluorescence, quenching, and rate constant curves at the bottom of the plot. This is likely because not all of the polymerized hemoglobin, not each hemoglobin in it, is not binding to the haptoglobin. Only 1 or 2 of the surface sites are binding to the haptoglobin, which is actually relatively promising because it would indicate a much higher relative effectiveness of the haptoglobin per hemoglobin in solution. So taking this, what we can instead now imagine is if we take the polymerized hemoglobin and it combines with the haptoglobin, it forms this larger complex. And looking at the right, what this should prevent is the polymerized hemoglobin extravasating out through the endothelium gap junction into the intima, where it would normally scavenge nitric oxide. To evaluate this system, we looked at the experimental hamster chamber window model. In this model, a hamster is outfitted with the metal clamp seen on the screen, and we can directly view the vasculature through this small window into the tissue. And we can measure things like the blood flow, the vessel diameter, and functional capillary density. In the experiment, what we did was first held the animal at baseline for 20 minutes, after which they would receive one of 3 treatments, either a saline, apohemoglobin alone, or the apohemoglobin-haptoglobin complex. The animals were then allowed 20 minutes to held another 20 minutes, and after we did a 20% blood volume exchange with a low molecular weight polymerized hemoglobin, and allowed an additional 20 minutes to observe any changes in the systemic or microcirculatory hemodynamics. First, looking at systemic hemodynamics, we found that the heart rate and the mean arterial pressure are maintained during the apohemoglobin-haptoglobin transfusions, which would be a good marker for its safety as a single therapeutic here. However, what we see much more interesting is when we go look at the microcirculatory hemodynamics. We found that the apohemoglobin-haptoglobin complex maintained functional capillary density, venule diameter, and arterial diameter during the transfusion of polymerized hemoglobin. This would indicate that the apohemoglobin-haptoglobin complex is effectively attenuating the hypertensive response that we would normally expect from polymerized hemoglobin. Taking all of this together, we can conclude that the apohemoglobin-haptoglobin confuture was effective at reducing polymerized hemoglobin-induced hypertension. Additionally, this is with a 20% isovolemic exchange, and that an independent administration of the apohemoglobin did not decrease the mean arterial pressure or heart rate, indicating general safety of the material. As a quick note, this was a preliminary study, and there are several limitations. This model was very simple, and it really does not approximate clinical conditions, typically where a patient would be in hemorrhagic shock prior to the polymerized hemoglobin transfusion, or in potentially other disease states, which will likely need to be more fully explored before translating this material into further application. As a final note, I would like to thank our various members of the lab, specifically Ivan Pyers. He would like to briefly thank some of our collaborators at The Ohio State University, the FDA, and UC San Diego for their help with some of the materials and experiments for these studies.
Thank you for taking the time to listen to my talk today. If you have any questions, please reach out and I'll answer them as soon as I make them. Thank you again and have a wonderful day.